Professor Emerita of Microbiology in the College, co-chair with Kelly Andrews, Senior Associate Athletic Director at the University of Toledo, of the Women and Philanthropy Education Committee. On behalf of that committee, I am pleased to welcome you to a joint event sponsored by Women in Philanthropy and the College of Medicine and Life Sciences alumni affiliate. The presentation is being recorded and will be posted on the alumni website early next week, so stay tuned. Also, please send your questions to our speakers using the chat function. Today's presentation is by Dr. Bina Joe on COVID-19 and metabolic dysfunction in hypertension. I am pleased to introduce our speakers. Dr. Bina Joe earned her PhD degree in biochemistry from the University of Mysore, India, and completed postdoctoral training at the Indian Institution of Science in Bangalore. She is a distinguished university professor and chair of the Department of Physiology and Pharmacology at the University of Toledo College of Medicine and Life Sciences. She is also the founding director of the Center for Hypertension and Precision Medicine at the University of Toledo. She is a fellow of the American Heart Association and a fellow of the American Physiological Society. She is editor-in-chief of the journal Physiological Genomics and an editorial board reviewer for the journal Hypertension. Dr. Cameron McCarthy earned his PhD degree in physiology from the Medical College of Georgia at Augusta University and did postdoctoral training initially at the Medical College of Georgia before joining Dr. Joe's laboratory. He is a fellow of the American Heart Association and currently holds the title of Dean's Postdoc to Faculty Fellow. He was awarded a National Institutes of Health K99R00 Pathway to Independence Award for July 2020 to 2025 that supports his postdoctoral training and transition to an independent faculty position. He is an editorial board reviewer of the American Hypertension and the journal Hypertension. One last reminder, please send your questions using the chat. Dr. Bina Joe will speak first, and I will turn this over to her now.
Thank you, Dr. Savicki. Let me share my screen. Okay. Can you see my screen? Can you all hear me? Yes, okay, good. First of all, thank you to Women in Philanthropy and the Alumni Association for hosting this virtual seminar. We are very grateful for the opportunity to present our research. And a big thank you to Women in Philanthropy for supporting us as we are one of the recipients of your awards a couple of years ago. Let's start with the topic of discussion today, COVID-19. It's coronavirus disease 2019. This slide is perhaps redundant to all of us because we are all experts at this time for knowing what this disease is. We are in the middle of a pandemic, which is very unfortunate. At the same time, we need solutions. So let's see where we stand with the science part of it in this talk. I'm not going to go over the first few bullet points. We all know it, except to point out that coronavirus disease 2019 is not only a respiratory disease, but it's being recognized as a vascular disease. Vascular disease immediately relates to what we're going to talk about today. It's linked to hypertension. And the last point here, long-term damage to organs is something new. It is not something that gets cleared and you're done with it. We are finding that we may have long-term damage to organs, in particular lungs and heart, and that is concerning. So more research on that has to happen, which is why it's very important to focus on this disease and its research. So what happens inside of your lungs is shown in this picture. The virus attacks the lungs first. what it does is it destroys certain cells called pneumocytes, which make a soapy material, which keeps your lungs healthy. But once that's gone, those cells are gone, the virus invades your lungs pretty quickly to the effect your lungs get flooded. So when we're breathing over water in our lungs, you can imagine it's almost like you're drowning and trying to catch a breath. So that happens very rapidly and the fluids fill up, and one can see that in a COVID-19 patient through x-rays. People don't even go to the part of x-rays, they pretty much die in a matter of 3 to 4 hours. So it can be very, very severe in some people. So the questions that we're going to address today are based on some of these data that I'm going to show you. If you look at the left-hand side, these are cases by age group. And on the right-hand side, these are deaths by age group. So if you glance over the pattern here for cases by age, is spread out through all ages. But when you look at deaths, you will see the graphs are longer at the bottom, where with age, there are more deaths. So question is, why is that? Why do older people have more severe COVID-19 and deaths due to COVID-19? The second data point we see pertains to pre-existing conditions. So if you look at this list from the CDC, people who are at an increased risk of severe illness are ones with cancer, chronic kidney disease, COPD, heart conditions, etc., etc. And people who might be at an increased risk are also listed here. Hypertension is here. Prior suspicions were that hypertension is here at the increased risk, but due to increasing knowledge, we now know it. They might be at an increased risk, and I'll explain why. And there are diseases not on this list, which means there are some with pre-existing conditions that may not contract COVID. So why is that? Why is it that only certain pre-existing conditions are more susceptible? Why not all ill health? People with ill health more generally susceptible. The third question relates to men and women. If you look at the left side, cases by sex and number of deaths on the right side, the cases, women are more. There are more women presenting with COVID-19. However, there are more men dying of COVID-19. Interesting, isn't it? So why is it that men are more susceptible to severe COVID-19, even if women contract it more? The last question relates to prevalence. And this is the chart from the Johns Hopkins database that we all follow, even our governor does. This is the world map, a world chart for how many cases have been there since March. There really hasn't been, waves, etc. It's been pretty steady in the world. That's interesting. And number of cases is on the rise as we speak. So why is it that it is dangerously fatal, for across the world? So we'll try and answer some of these questions. To get to the answers, here's some biology. The coronavirus uses a molecule called ACE2, which is an angiotensin converting enzyme 2 to enter our lung cells. Okay, so this is a very good molecule, and I'll explain why. And the virus uses this very good molecule in our body to enter our body. Why is it very good? Well, the good part of the story comes from what we know of blood pressure regulation. There is a system called renin-angiotensin system, and it looks too complicated, but just look at the bottom part of this. When you have a functioning renin-angiotensin system, you have two scenarios. One, you have ACE, which is angiotensin converting enzyme 1, and another ACE called angiotensin converting enzyme 2. If you have a lot of ACE, you make a lot of this molecule called angiotensin 2, which increases blood pressure. To balance this, you have this good guy, ACE2, where you have a lot of ACE2, you decrease blood pressure. So the regulation of blood pressure is maintained by how much of this mechanism is working and how much of this counterbalance mechanism is working. So without ACE2, you're likely to end up having very high blood pressure. Is the take-home message. So I told you ACE2 is very good. It lowers blood pressure, but it's also known to lower inflammation. So there are two good properties for this enzyme. And now you can see that when we don't have ACE2, which is the molecule used by the virus, and I'll tell you why it goes down when the virus binds to it. When you lower this, you increase blood pressure, you increase inflammation, and in the lungs in particular, what you have are these new type 2 pneumocytes. I told you these make the soapy material, which are surfactants. And once that's gone, you have very, very high rates of inflammation and fibrosis. And it can be acute death or it can be chronic infection and following. But if your immune system is not able to combat it, you pretty much die from it. So people with lower ACE2 are in trouble for contracting COVID-19. Okay. All right. So when do we have lower ACE2? Well, lower ACE2 is present in a hypertensive condition because of the two pathways I told you. The first one's working very high, second one isn't, ACE2 isn't, so you have very high blood pressure. It's also low in diabetes in the elderly with cardiovascular illnesses. So notice, all elderly may not get COVID because only elderly with cardiovascular illnesses will have lower ACE2. And that's the piece that is key for, from what we know, for how much one would get one would contract COVID-19. So there's this paper and many others saying that high blood pressure is linked to increased risk of dying from COVID-19, and it explains, the explanation goes by lower ACE2. So I was also going to tell you about how this virus uses ACE2 to come into our bodies and destroy the cells in our body. But what's more important to note is a dichotomy I brought up. I just told you that this molecule is what the virus uses. And I also told you that having lower ACE2 is a problem, which means it's counterintuitive, isn't it? If you have a lot of ACE2, virus should be able to happily bind. And if you have lower ACE2, why is it you are succumbing to COVID-19? Well, here's the story for that. If you have ACE2, virus can of course bind to it, but once the virus binds to it, this little cartoon shows what happens next. The virus binds to ACE2 and it's internalized, and it goes into these vesicles called lysosomes where the virus, the RNA molecule of the virus is released into the cell. So that's how the virus gets in to multiply. But the carrier, the ACE2, which is in green, gets destroyed. So when it's destroyed, you have lower ACE2. And if you don't have enough of reserve ACE2, whatever you have is already destroyed. Of course, the virus can't get in, but the virus has already gotten in. And so it's going to infect, and ACE2 isn't present. You can have more and more inflammation. And so you end up having very high late-stage inflammation and fibrosis and have COVID. Okay? So the key to remember is once the virus uses it, gets in, it destroys it, so you have inflammation to get the disease. So it's dangerous to have low ACE2 and no reserve. So increasing ACE2 is therefore beneficial. Okay. Now, where are situations where you have increasing ACE2? Certain antihypertensive medicines that we may be using as hypertensives increase ACE2. So if you are hypertensive, and if you are using some of the medicines shown here, these medications increase ACE2. So by logic, you may be protected from COVID-19 simply because these medications increase this good guy, increase ACE2. So let's look at the data. The same paper I alluded to, they looked at the death rate of hypertensives for using different kinds of medication. If you look at ACE inhibitors or ARBs, which are these medicines here, those who use these medicines have a lower percentage of death due to COVID compared to the antihypertensives that did not use these medications but used some other medications, the death rate is 9.8%. It's very early data, but it's suggesting that use of these medications may actually be protective for contracting COVID-19. And I told you that women have increased protection, right? Even if they contract the virus, they're not dying of COVID-19. Why is that? One of the answers to that question lies in our genes. This particular gene, ACE2, is located on chromosome X. And chromosome X is a double dose in women, as we know, and a single dose in men. So there is this genetic mechanism called X inactivation, whereby not all the genes on the X chromosome are double dose in women, So we get one of the X chromosomes randomly inactivated. So you only have one copy of the ACE2, but not so with ACE2. ACE2 escapes inactivation. What that means is all women have 2 active copies of ACE2. And men have only 1 copy of ACE2. So women have this very good molecule in double dose compared to men. When it's in double dose, of course, you have the virus coming into our bodies more because they can use this receptor. But when you have this double dose, you also have protection from inflammation. So even if the virus gets in, it can't do more damage because you have an anti-inflammatory property from this ACE2. That is why women have increased ACE2. The other reason is we have estrogens, which also increase ACE2, which I don't want to talk about today, but that's another point why we are protected, which means menopausal women may not have that benefit. They still have this benefit from the genetics part of it. The next point I'm going to go to is GI symptoms. Now, you have heard very recently, doctors are finding more patients with COVID-19 presenting with GI symptoms, loss of appetite, nausea, diarrhea, abdominal pain. This doesn't look like COVID-19, but doctors are paying attention to why that could be. And as scientists, we are paying more attention to why that could be. And here's why. As the Women Philanthropy organization knows through some of our previous interactions and the Alumni Association too, for some of my presentations there, our lab is heavily focused on studying gut microbiota. Now, it turns out gut microbes are an important feature for their association with ACE2. Why? Because of our work, and I'll tell you about it. But here's the first point to remember. ACE2, which is the good guy, is highly expressed in the gut where microbes reside. So far, the storyline was all in the lungs for attacking pneumocytes. But ACE2 is also present everywhere else, like the kidneys, the blood vessels. But why is it expressed in the gut where these microbes are reside? Nobody knows. So the question we posed is, does the gut microbiota have anything to do with ACE2? Anything is a very loose term. What it means is if it does regulate ACE2, we have a project. So we took it upon ourselves to address this question using what we call germ-free rats. We took rats that are completely devoid of these germs and colonized the rats with the germs for 10 days. So there were 2 groups, one without any germs, one with the germs. And then asked what happens to ACE2 expression in the gut. So we recently published this work. We were delighted to do so. One of our postdocs, Tao Yang, took it upon himself to look at the data we had. And here's the data on the right-hand side. This is the fold change of ACE2 in the gut in the germ-free rats and the germ-free conventionalized rats, the ones with the germs. Here you will see that ACE2 in the gut is lower when you have germs in the gut. That doesn't mean germs are harmful. This is a concept, proof of concept that germs in your gut may have something to do with ACE2 in the gut. What it doesn't tell you is which germs are beneficial, which germs are harmful, how is this relationship working when you change your gut microbiota. To do so, we need more research, which is an unanswered question, and so are many more questions. There is a lot more unknown about ACE2, particularly in the gut, and why people present with GI symptoms for COVID-19. Okay? And this is a busy slide, but I just wanted you to take home the message that there is action in the gut that is less researched and more clinically presented. So it's an important question that we want to address. So here in the university, during the lockdown, we brainstormed and came up with a project to address this. First of all, there are no good animal models with hypertension that also develop COVID-19. So we developed a new rat model. What did we do? We took the hypertensive rat that has the rat ACE2 gene, of course, on its genome, on its X chromosome. But the problem is these rats do not get the human SARS-CoV-2 infection, meaning they do not get COVID-19. Okay? So we have to solve this problem to have a model that would mimic human COVID-19. To do so, we engineered this rat to, first of all, get rid of the rat ACE2 gene and in its place, replace it with the human ACE2 gene. So now the virus can use the human ACE2 gene, a protein, to get into the body of a rat, right? So once we can infect this rat with the human virus, this now becomes a humanized rat model, not just for studying gut pathology, which is what we are interested, but also for testing other therapeutics and vaccines that may come along with many of the companies and academics working on this, right? First would be a preclinical model to test these. And if this rat gets infected, this would be the world's first model with hypertension and having the propensity to develop COVID-19. So wish us luck. If we do, Toledo will be quite famous for what its contribution is to the rest of the world. We do not have the data on infectivity, but our microbiology department has a fabulous biosafety level 3 laboratory, which is where we'll be trying this infectivity study with our microbiology department colleagues. So that's the latest from us. Hopefully the NIH will fund us for this project, and we will have more to come back with in the next few months here. Okay, so let me try to get the answers to what the questions we posed at the beginning of my presentation. Why do older people have more severe COVID-19? I know I gave you a lot of information, but focus on ACE2. Most of the answers are related to all the questions to ACE2. Older people have less ACE2 are the ones that are more predisposed, right? They may be less infected, but they're more vulnerable because their type 2 pneumocytes, not type 1, sorry, are killed and ACE2 levels fall further, and most of these lung cells get destroyed without ACE2. Okay? And also they have less immunity. That's another point that may not be more generalized here in my presentation, but I'm pointing to why pre-existing conditions with ACE2 being lower is something that doctors pay attention to. So why do people with certain pre-existing conditions present with COVID-19? Why not all? The same answer. The vulnerability for COVID-19 is not about general illness or weakness, but rather specific weakness with respect to lung protection via the ACE2 system. Why are men more susceptible? Well, we know women have 2 copies of protection from 2 copies of ACE2 by genetics. And as I alluded to, they have more estrogen, which has a positive effect on ACE2. Higher ACE2, although makes it easier for getting infected, you also have more reserve resistance to severe disease, which happens in women more than in men. And what makes severe COVID so fatal? The fact that this virus binds to ACE2 in the lungs, which we use to breathe. They lose their surface tension and floods the lungs with fluids, tips the balance for gas exchange within a matter of hours. So it can be dangerously fatal even before you get to the hospital because this step happens very rapidly. Lastly, why do certain COVID patients present with GI symptoms? We do not know the answer, but we were the first ones to present in the literature that there is a relationship between gut microbiota and gut ACE2, and further studies are to be conducted here at UT and elsewhere perhaps for understanding why that occurs. So what can we do? We are in the middle of a pandemic. There's nothing much we can do until science kicks in for developing vaccines, which is the number one feature that we would like scientists like us to present to the world. Secondly, I think we need to learn more about this ACE1, ACE2 system, especially in the gut, so we can avoid people thinking they have GI problems for something other than COVID-19. So that science is something we have to contribute to. And ultimately, we need to know that really there are no cures for viruses. Even the flu, we don't have a cure. We just have antivirals to slow down the viral replication until your own immune system can clear it. And supportive therapies where we could come in as an added advantage with COVID-19 if we find ways to increase ACE2 from what we have learned today in this presentation. What can the public do? We are doing everything we can. 3 things: social distancing, masks, and hand washing. This is highly preventative. So all groups of people urge people to do this until we can develop vaccines. And what can medicine do? What do doctors have? Doctors don't have much to do until we provide the answers for them to use as scientists. But for now, they consider ACE2 status based on your medical history before treating COVID-19. So they're paying attention to that factor. For example, if you're a hypertensive and you're using an ACE inhibitor and still presenting with COVID, they know your ACE status, ACE2 is great. They might look at other ways to treat the illness. There are many other ways that are particularly pointedly focused on the SARS-CoV-2, for which doctors try to use medications for, some of which are shown here. The virus enters the cell and does many things. So each step of the way, they're trying to prevent the virus from replicating. Convalescent plasma is something talked about. What that does is to have antibodies. So the virus has a mask basically, so it cannot bind to ACE2. It already is binding to the antibody. So that's the convalescent plasma story. And doctors use steroids for controlling inflammation because, as I told you, ACE2 is an anti-inflammatory molecule. If inflammatory molecules are low, you have to provide extra means to control inflammation, which is why they use steroids. And of course, remdesivir, that we know is an antiviral that's way down inside here, trying to prevent the virus from replicating further. I told you about many of these medicines, but I didn't allude to chloroquine. So what about chloroquine? My colleague and postdoc, who has been NIH certified now to do work, will talk to you about chloroquine. And Cam, the floor is yours.
Good afternoon. And first, I would just like to thank the Women Philanthropy Education Committee and the UT College of Medicine and Life Science Alumni Affiliate for this opportunity to speak this afternoon. And also thanks to Dr. Joe for that exciting intro into upcoming lab projects. So yes, what about chloroquine? As many of you may know, chloroquine is a classical antimalarial drug. To take us back a little bit to high school chemistry, chloroquine and its analogs, such as hydroxychloroquine, are diprotic weak bases, and its unprotonated form can selectively enter acidic vesicles within cells. These acidic vesicles are also known as lysosomes. Subsequently, chloroquine can become protonated and is unable to leave the lysosome. This is known as the Henderson-Hasselbalch law. So what does this mean in the case of malaria? You have the malarial parasite entering in red blood cells, and in the red blood cell, it starts consuming hemoglobin in the food vacuole. Food vacuole is another name for lysosome. As hemoglobin is degraded, you have the generation of toxic heme, and then subsequently this heme is converted to non-toxic hemozoinin. What we do is when we treat a malarial patient with chloroquine, it prevents this conversion from heme to hemozoin. And so therefore you have this toxic buildup of heme that subsequently kills the parasite and also the red blood cells. But we can make new red blood cells. The main thing is declaring the parasite. So in COVID-19, there have been 3 general mechanisms proposed by which chloroquine and its analogs act. First is it prevents viral entry into host cells by blocking endocytosis. The second mechanism is prevention of viral delivery of its RNA to the host's nucleus. And the third and final mechanism of action is pleiotropic anti-inflammatory effects. Interestingly, these anti-inflammatory effects of chloroquine are the reason why chloroquine has been approved to treat autoimmune diseases such as lupus and rheumatoid arthritis. Nonetheless, despite much hype, clinical trials have not supported chloroquine as an effective therapeutic in the treatment of COVID-19. This is in the context of both speed of recovery and also decreasing mortality. Additionally, some trials have been prematurely stopped owing to unintended side effects, including, but not limited to, cardiac arrhythmias, blood and lymph disorders, renal injuries, and liver failure. Now, many of these unintended side effects come about because chloroquine interferes with an important cellular process known as autophagy. Autophagy is our constitutively active catabolic process that's important for maintaining homeostasis via the efficient degradation of cellular debris. As you know, autophagy exists on a continuum. So when you, at one end of the continuum, if you have insufficient autophagy, such would be the case if you consumed chloroquine, you would have accumulation of undegraded cellular debris. This could lead to dysfunction and eventually cell death. On the other end of the continuum, if you exacerbate autophagy, you have basically the cell eating itself. And again, this can also lead to cell death. Now, the magnitude of autophagic activity is very much dependent on the models you study, whether that be human patients or animal models, the stress you impose or your intervention, and also the tissue in which you're measuring autophagy. This can change depending on the organ's requirement for maintaining homeostasis. Now, even though autophagy magnitude can change from tissue to tissue, the machinery it uses by activating autophagy is very much conserved, and it is initiated through conditions such as nutrient depletion or nutrient abundance. And although it is a very complex cascade, it basically converges on the synthesis of this double membrane lipid biolayer called a phagoform, The phagophore engulfs the cellular debris to be degraded in what is known as an autophagosome. And in the final step, the autophagosome fuses with a lysosome. The lysosome contains our acidic hydrolases, and these acidic hydrolases degrade the cellular debris that needs to be disposed of. Some of this goes out as excreted. Other parts are used as building block for new biomolecules. Now, the importance of this system to our cellular health is best illustrated with the fact that the 2016 Nobel Prize in Physiology and Medicine was awarded to the godfather of autophagy, Yoshinori Ishimi, for his discoveries of mechanisms for autophagy. Now, what does autophagy have to do with myself, who is a hypertension researcher and vascular biologist? Well, it's been well established that autophagy and relative vascular age are associated, and it has been proposed that decreased autophagy may be a novel index of vascular aging. So what do I mean by relative vascular age? Well, relative vascular age is working on the concept that vascular function deteriorates as we age. And in these disease conditions, you have a premature onset of this vascular aging. Now, vascular aging can encompass a number of different phenotypes, including endothelial dysfunction, vascular remodeling, stiffening, inflammation, or oxidative stress. And it has been revealed by a number of different studies that autophagic activity decreases with the onset of these phenotypes. In addition, it has been shown that upregulation of autophagy or reconstitution of autophagy is able to prevent vascular aging. This includes lifestyle modifications such as exercise or caloric restriction, which is well known to induce autophagy. You can have caloric restriction mimetics. So these are pharmacological agents that are able to stimulate the starvation state. And also you can have genetic transactivation of autophagy, have all been shown to prevent vascular aging and its associated phenotypes. Moreover, it has been shown that mice that have constitutively active autophagy through this ATG5 protein have an increased lifespan compared to wild-type mice, illustrating that autophagy is also essential for our longevity in addition to our vascular health. Nonetheless, despite this well-known association, the mechanisms by which autophagy decreases vascular aging and increases longevity remains the subject of much research, including our own. In addition, it has been revealed that autophagy is a key regulator of lipid metabolism and ketone body synthesis. And this brings us to our lab's favorite ketone body, beta-hydroxybutyrate. BOHB is predominantly made in the liver in response to energy deplete conditions. So what we have revealed is that autophagy is a novel regulator of BOHB in the liver. And our second novel discovery is that once BOHB is released into the circulation to serve as a nutrient source, that BOHB can cause potent vasodilation in arteries from normotensive and hypertensive animals. And this is really striking and really pushes the field forward. And we're really excited about the potential therapeutics in terms of harnessing this mechanism in the future. So to summarize this work briefly, We believe we're working with two important mechanisms. The first is that physiologically, increases in liver autophagy would increase beta hydroxybutyrate, and this can decrease premature vascular aging and hypertension. And on the other hand, pathophysiologically, when you have decreases in liver autophagy, you would have decreases in beta hydroxybutyrate and increases in premature vascular aging and hypertension. So I don't have to belabor the point to this group of how important blood pressure control and preventing hypertension is important for our quality of life and living a long and healthy life. As we know, hypertension can significantly decrease both of these. And with that, on behalf of Bina and myself, I'd like to take this moment to thank our lab for their continued support, our collaborators additionally for their support, and our funding agencies, including the Women Philanthropy group. And with that, Bina and I would be happy to take any questions. Thank you.
So at this time, you can get off the presentation and go into the chat box to see if you have questions.
Okay. So reading through the questions. Question for Dr. Joe and Dr. McCarthy. There is great interest in when a vaccine or vaccines may be available widely. From a scientific view, when do you think this is most likely that a vaccine would be available to the general population in the US? And in your opinion, will people be resistant to receiving vaccines that have been moved so quickly through the regulatory process? You're welcome to go first, so I can comment after.
Okay, so thank you for that question. We are, for all intents and purposes, part of you guys, the public, right? Just a little bit more insight into the science of vaccine development. It takes a long time, a few years, to get a proper vaccine going. That's from the scientific point of view. But there are new ways to develop vaccines. For example, targeting RNA, which is what one of the companies is trying to do. And those are newer methods. So there are new methods that are being tested, which could result in faster vaccines. But the process of vaccine development has to go through clinical trials, which takes time. The danger of not going through the proper channels or proper mechanisms is we get vaccines that are not functional. So political groups have their views, the general public has their views, but as scientists, my own personal point of view is give it a few years. And to expect that to be a few years is reasonable. Secondly, mass production of the vaccine itself. There are countries like India that I know of that most of the vaccine production to about 70%. So the workload for that to scale up production for people in the US to have vaccines will also take time. So this issue of many months can be variable from 6 months from now. We have a vaccine that can be tested, but it has to be mass produced, maybe a few more months. Give it a year or more if the initial ones are not successful.
I would also like to add that the NIH director, Francis Collins, and other prominent scientists within the vaccine development and manufacturing industry have come out and publicly stated recently that they will not be pressured into prematurely releasing any sort of vaccine, and that all vaccines that are eventually approved will go through all the rigorous regulatory processes that all vaccines go through. So we have been reassured by the top health leaders in our country that this is going to be safe and effective for use by the public.
On a positive note, we can be sure there will be a vaccine. The timeline isn't what we expected to be.
No, that was the question.
Okay. If people have more questions, we have perhaps 5 more minutes in the chat box. At this time, also, I would also give my phone number in case people want to call in after the hour and have specific questions. Happy to by typing my phone number on the chat box as 49.
Should I go ahead?
340.
Cameron, I think you can also look in the Q&A box.
Yeah, I think there's a couple things.
Oh,
in
the Q&A box. Sorry, I'm looking at the chat box.
there. Yeah, there were a couple more.
Sorry about that.
Okay, when they're done. Okay, thanks.
Oh,
Yeah,
increase this. Oh, here we go. Sorry about that. I have, from Eric, I have high blood pressure, but take lisinopril. Does this increase or decrease my risk of COVID?
It should decrease your risk because lisinopril is an ACE inhibitor. So, by what you have learned today, you should be safer than somebody else who's not using lisinopril and effective. How about that?
so that's a good answer.
Second question from Catherine. Are all countries reporting cases and deaths accurately?
Well, as scientists, we cannot answer that question. We can talk to you about lemon and then we can have a debate or discussion after this hour.
I certainly have my opinions, but yeah, they're not scientifically backed up. So I'll.
I think we are trying to be Dr. Fauci's here, trying to stick with the facts of science.
Now, what would be a good food to eat to prevent me from getting COVID-19? In another word, how can I have a strong immune system in order to prevent me from getting COVID-19?
Best answer for that is obviously we don't know is the best answer, but from knowing that anti-inflammatory molecules help you to keep diets that are rich in anti-inflammatory molecules, or foods rich in antioxidant, anti-inflammatory, have to be careful, somewhere in that group would be good. By no means am I promoting nutritional therapy for COVID-19. It's an impossible situation. But fruits with antioxidant properties, anti-inflammatory properties of certain spices, etc., in the literature, so they may help. And again, knowing that chloroquine, which is also anti-inflammatory, did not work, and knowing ACE2 is anti-inflammatory and it does work, there's a difference between all anti-inflammatory molecules to work for you for COVID or not. So there is no data to support anything in the diet to help you prevent COVID-19. On a general note, having your immune system heightened with probiotics, etc., whatever you feel good with to increase your innate immune responses is a good idea.
I'll just add that following the food pyramid, if you remember that from health class in school, that's always a good rule of thumb for healthy fruits and vegetables and relative abundance to consume for a healthy immune system and to prevent like the flu and now COVID. So a healthy diet, a balanced diet, rich in fruits and vegetables, like Bina suggested, is probably your best immune defense from that respect.
I can also add from what we learned from Dr. McCarthy's presentation, not just diet, BOHP is a huge molecule increase with exercise. So if you can exercise more, your blood vessels are more dilated, your blood pressure can be more controlled. By that means you may be not in the predisposed group, right, for developing COVID. So try to exercise more if you can. It's a good thing.
Next question is from Anita. I understood ACE2 for women who are in menopause may not get whole benefit. What about women taking estrogen or other related menopause drugs?
Should work. If your estrogen is high, it's known to upregulate ACE2. You should have the benefits of estrogen. But as a woman, you always have two active copies of ACE2 working. Whether you're in menopause or not, the genetics part of it is still working. The estrogen part of it is notched down in menopause. So you don't have to worry about it, the estrogen going away. If you're on an estrogen supplement, you are raising that also in addition to your double copies. So you're good.
Jane asks, will any vaccinations be without formaldehyde or phenols? I wouldn't know that answer, sorry.
Probably not. But those are out there once we make the vaccine, how to package it. It's like asking, can I have a pill or a fluid? What the composition of the vaccine is. So we don't know how to answer that question at this time. So we don't have a vaccine yet.
Along with hydrochlorothiazide for hypertension after breakfast, is it okay? Sorry, I don't understand. Along with hydrochlorothiazide for hypertension after breakfast, is it okay? Oh, is this related to, oh, sorry, it's split, that's why. Currently, I'm taking Losartan for hypertension, along with hydrochlorothiazide for hypertension after breakfast. Is it okay?
Yes.
If your doctor prescribes
Yes.
that.
Yes.
Does it help that Oxford has been studying common cold for years? Will they be first vaccine?
Well, first vaccine is anybody's race. Astra has it, Oxford has it. There are many companies working on it. Somebody may be a dark horse coming forward later. So many companies, including the Bill and Melinda Gates Foundation, they're funding a few. It's just very nice to have many competitors. I don't know who's going to win the race or not. I wish there are many who will come up with different vaccines, different approaches. So you have an RNA vaccine, you have a protein vaccine, yada yada.
Sarah. Question was asked about efficacy of vitamin C, and also what about effectiveness of chloroquine in view of many discussions on social media?
Definitely, Dr. McCarthy has shown you that chloroquine doesn't work. Vitamin C, there's no test yet. Again, it goes into that component of antioxidant properties or anti-inflammatory properties. Vitamin C is one of those. So using those can increase your innate immune response. So by definition, it should be good. It may not prevent COVID-19 totally because of all the other factors surrounding it for age, sex, age-2 copies, and age, etc. I mean, what do we have?
Just a couple.
Okay.
Just in view of the comment by Sarah about many discussions on social media, just despite what's posted on social media, the
Have another
chloroquine was
question?
not shown to be effective in
If
randomized
not, I'd like to thank our
placebo-controlled.
presenters, Dr. Bina Joe and Dr. Cameron McCarthy, for helping to demystify COVID-19 and hypertension for us. Thank you also to Cynthia Sawicki, Kelly Andrews, and the Education Committee for organizing this extremely timely and informational event. And thanks to the College of Medicine and Life Sciences Alumni Affiliate for joining us in presenting this event today. We're so glad to have members of Women in Philanthropy that lead to a better understanding of how the university is helping to improve lives across Northwest Ohio and beyond. A reminder that this presentation will be posted online early next week for those who would like to view it again or who were unable to attend today. It will also be posted on the Alumni Association's website, as well as our Women in Philanthropy Facebook page. We are very much looking forward to the time in 2021 when we can gather together in person to support our university, our students, and the entire Rocket community. Enjoy the rest of your day and go Rockets.
Thank you.
Oh,
Good night,
Sarah,
thank you.
you could-
Good night, good afternoon.
Oh, okay. All right, we are here if people want to ask a question.
Yeah, we got a couple minutes. We can.
Okay, do you have any more questions that came
There
in?
was a couple more questions.
Okay, we'll go ahead with that. I guess I jumped the gun.
No problem. All right, just trying to scroll back up.
We have about 4 minutes.
Any diet modifications that help the immune system from Michael? Just like Bina mentioned, a balanced diet, rich in fruit and vegetables, I think is your best bet to keep your immune system strong. Is there a test to determine if you have high or low ACE2 levels? That's a good question.
That's a good question. We can test it, but I do not know if it's approved by the medical community to do so for your insurance to pay. But definitely there are tests we can do to determine your ACE2 levels.
And susceptibility to COVID may depend on where the ACE2 is expressed. So necessarily testing your blood levels of ACE2 might not be indicate how susceptible you are to the disease, unfortunately, because as Bina mentioned, it's important the levels in the lungs, and what we are showing is the ACE2 levels in the gut also. How can I know if someone has COVID-19 or the flu? Thank you.
For COVID-19, there is a specific PCR test for the viral sequence and an antibody test for an ELISA. But for the flu virus, it's a very different sequence. So you will have custom designed tests for both viruses differently. Not for that matter, all viruses have different sequences. So there are custom tests for both or more, dengue, etc., that you can test for.
All the rest of mine are comments. Ben states, as a long COVID-19 survivor, after effects from COVID are many. My case, it's neuropathy, ulcers, partial paralysis.
That is very sad.
Hope you make a speedy recovery, Ben. Comment by Lance, antihypertensives are more effective if taken at bedtime. That seems, does anyone else have any more questions or comments?
Well, if not, I think Dr. McCarthy has posted my number. Feel free to call me. I'll be very happy to address your questions. Thank you so much. Okay.
Thank you. And yeah, we're.
Dee has something. Dee, are you trying to say something?
We're muted.
Yeah, we can't hear you.
Got everything covered.
Okay. And also we can be found via the Physiology and Pharmacology website. Our emails are there and office phone numbers if you would like to contact us. Please feel free.
Thank you very
Thank you
much.
all.
It was
Thank you,
very
everybody.
informative.
Bye-bye.
Bye-bye.